I think we're going to start. So thank you very much, everyone, for coming to our Innovative Discovery Series seminar. Before introducing our speaker today, let me give you some housekeeping information. Please make sure you sign in. The sign-in papers are in the back. Please RSVP so that we can better prepare for the next session. Online participants, please keep the mics muted unless asking a question and keep video turned off. Use the chat feature in BlueJeans if you have any technical questions. Our next seminar will be Qualitative Methods, Tools for Understanding Patient, Provider, and Community Experiences by Dr. Rosemary Presso on February 19th. And then that will be followed by the use and misuse of statistics in medical research by our own Paul Cole on March 4th. So please see the full schedule on the ACCEL website, and know also that all the previous presentations are available on our website, DE-CTR.org. So I'm really delighted to introduce Dr. DeLeon today and that she accepted to come and give a talk at our seminar. Dr. DeLeon is currently the Trustees Distinguished Professor of Biological Sciences at University of Delaware. He obtained a Master of Sciences at University of the West Indies and in Jamaica, and a PhD from University of Western Ontario in London, Canada. She also was a postdoc at McGill University in Montreal, in Canada. And she's had multiple awards over the years. For example, in 2009, and it's only one that I selected among a long list, she obtained the Presidential Award for Excellence in Science, Mathematics, and Engineering Mentoring. It is an award administered by the NSF and awarded only to 10 individuals or organizations per year nationally. And in 2015, she was honored at FICAPA, a selective organization that celebrates scholarship and academic success. Dr. DeLeon has been extremely successful in gaining grant support over the years, and she's also an exceptional mentor and has supervised and mentored multiple graduate students, faculties from assistant professors to associate to full professors. She's also a very prolific author and has published more than 90 peer-reviewed publications and several book chapters. And she also has patent applications that have been issued or are pending. So welcome, and we are really excited to hear about your research.
Thank you very much for your generous introduction. Kind of nice not to have to hassle to the airport to get to where you're going, but then there's a parking. Anyhow, I'm very happy to be here today to tell you about some of the work we've been doing in our lab. Quickly to begin, I just want to explain sperm mother, emergency will be in the data ECA 10 minutes, center 3 to center 3. Sperm mother, emergency will be in the data ECA 10 minutes. Okay. Yeah, I just want to, I'll begin by looking at the requirements for fertilization in vivo. And when these requirements aren't met, it can be infertility, which unfortunately is an increasing problem. Then I'll spend some time talking about the role of genetic factors in unexplained male infertility, because I'm an andrologist and so I'm focused on the male. And then looking at some of the current options that we have with ART and some concerns, of course. And reproductive geneticists and andrologists have certain goals to try to address these concerns. Then I'll talk about some of the work done in my lab. I'll focus on 2 protein families I've worked on over the last 15 years. And then end my talk by talking about the application of insights learned from nature to improve the success rate of ART. Testicular maturation of sperm occurs when a round spermatid becomes transformed into a polarized cell. In this polarized cell, the nucleus is highly condensed, and as a result of this high degree of condensation, it is synthetically inactive. Therefore, all the proteins that it gains after leaving the testis are acquired from the environment in which it passes. And so when it gets to the epididymis, it gains proteins on the membrane. And then it gets into the female tract, and it also gains more proteins. And it goes through its final maturation period, which is called capacitation. Of course, the business end in the female tract is the oviduct. And the oviduct is a bidirectional route where sperm travels
from the uterus into the ampular junction, the ampular isthmus junction, where the sperm will meet the oocyte. And the other direction, of course, once fertilization has occurred, the zygote and early embryo travels the other direction towards the uterus for implantation. So it's a bidirectional route. But there are 3 points I want to make about the oviduct. One is that its secretions interact with the gametes. Secondly, is that the gametes modulate the environment, the oviductal environment. And the third point is that the early embryo spends about 2 to 5 days in the oviduct. This makes the oviduct a very, very important organ. Of course, when the sperm travels, it must meet the egg, and that is an absolute requirement for fertility and for reproduction and the survival of our species. It must travel, and it must recognize the oocyte. It must penetrate barriers, and there are 3 formidable barriers that exist. There's a barrier on the cumular cells, and the zona pellucida, and the perivitellin space. And by virtue of enzymes on the surface of the sperm, it's able to break down the extracellular matrix in these barriers and get access to the oocyte. So once the barriers have been penetrated, there's fertilization and the delivery of the genome to the zygote. Now, the inability, the absence of any of these events can lead to infertility, and this is the inability to conceive a child or difficulty in conceiving a child for a period of 1 year or so. It is indeed a worldwide problem. About 15% of all couples face this problem. It leads to sometimes psychological problems. And it's been estimated that about 50% of these cases result from a male factor and 50% from a female factor. And because I'm from Delaware, I use the blue and gold to represent these. What I want you to see is that there are a number of causes in male and female. I'm focusing essentially mostly on the male causes. And what you see is, before I mentioned that there's poor sperm quality is a major factor. But I want you to see that genetic factors play an important role in both the male factors as well as the female factors. I mentioned that the problem is on the rise and has become a serious public health problem. Just to quote a few studies, in Copenhagen, between 1938 and 1991, there was a significant decline in semen quality due to environmental pollutants. And between 1960 and 1990, there's a 28% increase in men with oligospermia, or low sperm count. Going back to this country, there was a study done in upstate New York State University where they showed that the use of laptops reduced sperm counts. And of course, lifestyle, hot tubs, sauna, pesticides, and of course, obesity. So our present lifestyle has been contributing to an increase. In 2002, the National Survey of Family Growth from CDC reported that 7.5% of all sexually experienced men reported a visit to help with conceiving a child, and that equates to about 3.3 to 4.7 million men. So overall, there's a 1 in 6 of all couples face this problem of subfertility or infertility. Just to show you again how worldwide the problem is, in India, 12 to 18 million couples per year are diagnosed with infertility. And in the last 30 years, the sperm count has declined from 60 to 20 million per male due to xenobiotics, which are foreign chemicals emanating from the chemical industries. Of course, environmental heat has been a problem as well because it increases scrotal temperature. And a study, which I found quite amazing, from the UK showed that 1 in 5 18-year-old boys can be classified as being subfertile on the basis of semen quality, that is the motility and morphology of the sperm. WHO, World Health Organization, has reported a worrying recent decrease in male fertility, 50% decline in sperm counts in 15,000 men in 21 countries in the last 25 years. And this is an infertility time bomb, which is almost as important as, in my estimation, as climate change. And of course, the single most common cause of infertility is the sperm defects. And so let's look at the sperm. It is a remarkable motile machine. It is designed for traversing the female reproductive tract and to undergo a series of complicated reactions. As I mentioned before, it's a unique polarized cell devoid of any synthetic machine. It has no ability to synthesize proteins and very little cytoplasm. So it needs a lot of protection. It needs a lot of in its travel, but doesn't have all the scavenger enzymes to protect it from harmful products. And then, of course, since there's very little cytoplasm, most of the proteins are membrane proteins, and in a way, provides easy access for study. Just as important, complex of the structure is also its method of formation, occurs by a process called meiosis, two cellular divisions, and it's been estimated that about 50% of the 20,000 genes we have in the genome are expressed in the testis. That's a half of the genome expressed in the testis. And 50% of the testis expressed specific genes are devoted to testicular sperm maturation function. I remember I mentioned early on that going from a round spermatid to a polarized cell, that is a testicular maturation, is required about 16 stages and involves a large number of genes. Of course, mutations in any of these genes could negatively impact their function. Our lab was the first to discover that a mutation in a gene called SPAM1, and SPAM1 stands for, sounds like a food item, but it's not. It stands for sperm adhesion molecule 1, and it was discovered in our lab that when there are mutations in that gene, the sperm are unable to penetrate the extracellular matrix or the pre-formidable barriers that exist. Consequently, there is a reduction in litter sizes. Turning to humans now, we know that 75% of all the cases of human male infertility result from causes are known, and they've listed here, involve varicocele, genetic factors, and so on. But I'm focusing on the 25% that is idiopathic. We don't know what's the cause there. But it's been estimated that significant fraction of that 25% involve genetic factors. And we do know that there are both chromosomal factors as well as genes that are involved. In terms of chromosomes, numerical abnormalities, if we have a loss or a gain of a sex chromosome, that's associated with azoospermia, the absence of sperm, or oligospermia. Similarly, if we have a structural deletion or of the Y, we have the same azoospermia. In terms of genes, it's well known that the cystic fibrosis gene has a variant that affects the bilateral absence of the vas deferens, and these males are sterile. So then a lot of the information we now have about the genes involved have come from studies in mice where we're able to knock the genes out. When we knock these genes out, we find that they're genes essential for sperm tail formation, for energy, for metabolism, for head condensation. I told you head must be condensed, for removing the cytoplasm, for head cap formation. All of these genes, these processes depend on specific genes, and when those genes are absent, the sperm are not able to function normally. Now, the idiopathic cases, the cases that we don't know of, and some that we others know, have been effectively treated by ART. And this is the techniques that allow one's own biological children to be born despite the difficulties. To date, about over 5 million children have been born by IVF techniques, in vitro fertilization, or ART, since 1978, when the first case came on the scene. That was Louise Brown, as a result of the pioneering work of Robert Edwards, and he received the Nobel Prize for that, I think, in 2010. Today, there are three major forms of ART. First is intrauterine insemination, where you just inseminate the sperm in the uterus. When this is really good when you have low sperm counts or motility is affected. Then there's the in vitro fertilization, where you have a population of sperm incubated with the egg in a dish, and this compensates for motility defects because you've done the motility for sperm. And then, so males with oligospermia also benefit from that. And then the last technique, and the one that's most predominantly used, I should say, is the ICSCI, which stands for Intracytoplasmic Sperm Injection. That has only about a 32% success rate. Now, if you look at IVF, the in vitro fertilization, it's quite similar to natural insemination in that the three formidable barriers I mentioned earlier on, the cumulus cells, the zona pellucida, the pervitellin space, are in place, and a microdrop of insemination is placed with the oocyte. They're incubated, and so there is some natural competition that normally goes on in the case of in vitro fertilization as occurs naturally. So there's sperm competition because the filters are in place. Now, on the left, there's ICSCI, and ICSCI involves just simply the technician chooses a sperm, and she chooses a sperm on the basis of its morphology and its motility. It's like a beauty contest. She chooses a good-looking sperm. And she knows nothing about the DNA or the proteins in the sperm. Nothing about all the proteins, but she chooses a good-looking sperm, and she injects that sperm. So note that for ICSCI, all of the natural barriers are removed because they have to remove these coats, outer barriers, and they do it with enzymes. One of the enzymes is the enzyme that I discovered. It's the SPAM1 enzyme. And so in the terms of both ICSCI as well as IVF, they all bypass the natural maternal gamete interaction that I showed you earlier on in the oviduct. Those are all bypassed. And so we now know that there are small but significant increases in the risk of birth defects that result from ICSCI. And long-term effects, their potential long-term effects, health consequences that we don't know a lot about because we've only been doing ICSCI for 20 years, so we don't know the projected. So it has definitely revolutionized treating male infertility, or treating infertility as of the field of infertility. But despite its indisputable efficacy, the efficiency is quite low, 32%. You wouldn't give it, if you're a student, you would send your kid home if you get 32%, right? You would, little spank in there with you or something. That's pretty low. And the potential for the long-term and short-term consequences, as I mentioned, we only had it for 2 decades, so we don't know yet. But what we do know from the short-term consequences is that there is an increase in genomic imprinting disorders in children born with ICSCI. There have been 3 reports. I've given 2 here, one by Cox and one by Lavergne. But we do know that there is some effect. And we also know that generally epigenetic factors are just as important as genetic factors. And epigenetic factors involve DNA methylation. And we can, when the technician chooses a sperm, she knows nothing about the methylation pattern of the DNA in that sperm. Similarly, we now know that there are RNAs in sperm, that these RNA play a regulatory role. So the current technology does not allow the detection of these factors, as well as DNA packaging and so on, that are really important for embryogenesis. There are other disadvantages. There's the expensive, it's about $12,000 per cycle, which means that limited availability to those individuals who are not very well. My daughter had her girlfriend mortgaged their house, get 3 times to get to be able to do it. It should not be. We should really do better than that, and I hope we are trying to do better than that. And of course, it's invasive. So additionally, what's being done in art is you're treating patients without an understanding of the specific effect. So you're treating the symptoms. It's a symptom treatment, not really the condition. So in order to make breakthroughs in this field, we have to require to have ongoing studies. And andrologists and reproductive geneticists have 3 goals. They we really want to identify relevant genes that can be diagnosed and assessed for mutations in men with or couples with idiopathic infertility. And for this, we need molecular research tools. We also need to develop a rational drug-based treatment for specific pathologies leading to male subfertility to circumvent the symptom-based treatment that now exists. And we also need to increase our understanding of gamete biology and molecular clues about the mechanisms that mediate maternal genotype embryo interactions. So what this does is requires information or the deeper understanding of genes and proteins that are involved in sperm function. Our lab currently works on two groups of proteins, or over the past few years, we've worked on two groups of proteins. First, they're membrane proteins, because those are what mostly you find on sperm. And they're coded by a variety of genes. And I'll spend most of the rest of the talk telling you about this and how we can apply what we've done. The first family, family of related hyaluronidases, and hyaluronidases are enzymes that break down the hyaluronan around the extracellular matrix of cells. And SPAM1, I mentioned earlier, is the best studied hyaluronidase. And then we'll talk about some novel players, which are more recently found. These are involved in handling calcium. Calcium is very important for fertilization, and there have to be very peaks of calcium, and it has to be brought back down to basal levels. And so PEMCA and its interacting partners we'll be talking about, because that plays a role in regulating calcium. So turning to SPAM1, I mentioned it's a sperm adhesion molecule. It's a multifunctional protein in that it in addition to breaking down the extracellular matrix, in addition to its enzyme activity, it also has an egg binding activity. It will bind to the zona pellucida as well. And it's conserved in every mammalian species that's been studied. It tells you that it's important. When everything is conserved in every mammal, it's important. In PAM1 is called, in some species, pH20. It's called 2B1 in rats. In cows, it's called P80, and all other names for it. And, it's a lot of confusion in the literature. But Michael Ashberger said it best when he says that biologists would rather share a toothbrush than share the name of a gene. And that happens all the time. They have the same gene, but everybody calls it, they find it in the pig, they call it that. In the cow, they call it that, and it's just really confusion. So I do concur with Michael Ashberger. So our discovery of SPAM1 was really exciting, because we found that when we took the sperm from the top of the epididymis, it had far less of the SPAM1 on it than when we took it from the bottom of the epididymis. You notice that you can see some SPAM1 on the tail here. You can't see any on the tail there. You can see that this is much darker, much lighter. And so remember, I told you that when sperm leave the testis, they are synthetically inactive. So if at the bottom of the tract they have more SPAM1 than at the top of the tract, it must have come from the tract. And it must have come from the secretions. And we were the first lab to discover that. And we actually went on to show that the same thing exists in humans. We were fortunate enough to get some material from males, and we found that this protein is expressed in the environment, and it's also picked up by sperm as well. So here is the tract, the epididymal tract, and here is the top, and here is the bottom. When they're at the bottom, they have a lot more than when they're at the top, which means that on its way down, it collects the protein. And the protein, as I mentioned before, it is multifunctional, and that it is delivered to sperm. We've done experiments in which we've taken mice that have no SPAM1, they have the gene knocked out, incubate them in the fluid in the oviduct, in the epididymis, and they've picked it up. And interestingly enough, when they pick it up, it increases the fertilizing competence. And so we know that this is a part of the process. Of course, our next question is, does the oviductal fluid contain and deliver SPAM1 to sperm just as the epididymal fluid? And the answer was yes. We found it not only in the epididymal fluid, but we found it in the uterine fluid, We found it in the ejaculate. It comes from the prostate. So along the entire journey, the sperm is able to pick up SPAM1. And I'll show you that it also picks it up in the oviduct as well. But before I show that to you, let me tell you exactly where SPAM1 is carried in the fluid. It's carried on membrane vesicles. There are lots of tiny membrane vesicles in the fluid that you can separate by ultracentrifugation. And they carry SPAM1. These are these vesicles that you see, the TEM. And here you see labeling, immunolabeling on these vesicles. And these vesicles are found in the epididymis. They were called epididymis. They're called epididymosomes. We found them the first person in uterus, and my PhD student was very happy to call them uterosomes. And prostosomes were found in 1978 by Ronquist. And the next question we ask would be, are these vesicles found in the oviduct fluid? Before I tell you whether or not they're found in the oviductal fluid, let me tell you how these vesicles are formed. These vesicles are formed by two mechanisms, one called the apocrine secretion and one called the multivesicular bodies. We'll just focus on the apocrine secretion because that is the one that is involved in our reproductive area. These vesicles, or extracellular vesicles, are pleomorphic in that they have different sizes. They range from less than 100 nanometers up to a micron in diameter. They act as a mode of communication between cells, and they I mentioned the two pathways. Now, the pathway we're going to focus on is the apocrine pathway, where the epithelial cells form these blebs. You can see those blebs on the epithelial cells, and these blebs are released. And when the blebs are released, the blebs release these exosomes. So that's the apocrine pathway. We, of course, did some sections in the mouse oviduct, and after we perfused the female, we did sections, and using TEM, you could see we detected the vesicles inside too. Some are small and some are large, and these are the cilia that are responsible for moving the egg down. And we also detected these blebs that had many of the little vesicles to be released. And if you look at the next slide, you'll see a very nice one. We did some tomography. If you follow this arrow through all these 6 sections of the oviduct, you can see here, you can see here, over here, there. You can see there, you can see there, you can see there. So that is one of the bleb that we actually caught in action. And that tells us that the formation of oviductal exosomes occurs via the apocrine pathway, where blebs are released from the epithelium. So how do we collect these exosomes to be used? And I should tell you, this last graduate student who graduated last year, she collected them by ultracentrifugation and then used TEM with negative staining to identify them, and looked at biochemical markers that are known to be found on exosomes. One very critical biochemical marker is a tetraspanin called CD9. And all exosomes that are found in all the body biofuels have CD9 on it. And we were able to show that CD9 was present by Western as well as by immunolabeling. We had the pleasure of calling them oviductosomes. And so the question is, after we discovered oviductosomes, one group in Paris in France and the other in Spain working on bovine and pig, also they have a lot more material to work with than I do with my little mice, but they discovered oviductosomes there, and interestingly, they found that SPAM1 was found on it. So we now know that SPAM1 is carried in the oviductosome. So quickly, to give you a summary of what we've done so far, is that I've talked about SPAM1 is a multifunctional sperm protein that penetrates the egg. Mutations in that protein in mice, are associated with reduced fertility. In addition to that, we found that SPAM1 is secreted in both the male and the female reproductive tracts, where it can be added to sperm to increase the fertilizing competence. And I didn't have time to tell you that although most of the SPAM1 is found in the vesicles, some SPAM1 is also found in the supernatant, in the fluid part, because SPAM1 is a GPI-linked protein. It has a lipid anchor that can hook it into the membrane, and it's hooked into lipid carriers. So one lipid carrier is called clusterine, and it's found with clusterine as well. But for this talk, I won't focus much on that. But just simply to say that one of the translational impact of our work so far is that we have identified specific conditions in which not only vesicles, but membrane-free SPAM1 and other GPI-linked proteins can be transferred in vitro, and that's what one of the patents that you mentioned is on. So this is really important, particularly for domestic animals, where all of the breeding, animal breeding, is done by artificial insemination, and most of the sperm are cryopreserved. And cryopreservation is known to destroy membrane proteins. So now we have a way of putting them back on and we have some experiments online lined up for that for domestic animals, and of course the ultimate would be to get to humans. So we turn now to the second group of proteins, and this is the plasma membrane calcium ATPase. This is PEMCA. It is a major calcium efflux pump in the body, and it's not testis specific. It's found pretty well throughout the body. Not throughout, but it's found in the brain and it's found in other places. By the way, a lot of genes that are expressed in the testis are also expressed in brain. Not sure the connection. But we find that this PEMCA4 is very pivotal in fertilization. It's a regulator of sperm motility. If it's not there, when you knock it out, sperm are immotile and the males are sterile mice. And the reason is that there's calcium spikes are required for capacitation, which is the final maturation of sperm in the female tract. It's required for the hyperactivation that sperm undergo just when they see the egg. They undergo this very asymmetrical movement. And it's also responsible for the acrosome reaction that must take place before they can actually get inside the egg. So three very important events require high levels of calcium. Once those have occurred, the calcium must be brought down to basal levels because calcium is toxic in elevated level. And that's where PEMCA comes in. PEMCA effluxes it out. PEMCA puts it out. And so in the absence of PEMCA, there is the loss of sperm motility and male infertility. Now, we were pretty happy when we discovered PEMCA because we'd worked for many years with SPAM1, and when SPAM1 was knocked out, there was no effect. We said, how could such an important protein for fertilization not impact fertility once it's knocked out? So we were pretty, it was hard to get funds from NIH. When NIH wants you to get infertility, we want our families to be, So we were pretty happy with PEMCA. And of course, the reason that the absence of SPAM1 does not cause infertility is that SPAM1 has some family members that compensate. They're the hyaluronidase 2 and hyaluronidase 3 are two family members that when SPAM1 is knocked out, they fill the place. I don't want to call the word redundant. I don't like the term redundant genes because each gene has a specific function, but they fit in. Well, there's nobody to fit in for PEMCA4 in the male, and so you have infertility. Of course, we went on right off to look to see if PEMCA4 was present in the epididymis, and if it was there, and if it was there in the epididymis and it was being secreted, well, we wanted to follow that up, and we did follow that up. And before, I should tell you a little more about PEMCA1, show you where it's located. It's located up over the head and on the mid-piece in human sperm, and on the mouse sperm, it's over the head as well, and on the proximal principal piece. PEMCA is a transmembrane protein. It goes across the membrane, unlike SPAM1, which is a GPI link, which is hooked into the membrane with a lipid anchor. This one is transmembrane, so you have a part of the protein inside the cytoplasm and a part outside where it ejects the calcium. So this is in terms of the protein. It not only, in terms of PEMCA's function, not only does it eject calcium, but it also, when it ejects calcium, it brings in one molecule of hydrogen. So it regulates pH as well. And as I told you before, the knockout animals are sterile. So we spent a lot of time working on why does the absence of PEMCA cause immotility of sperm and ultimately infertility. And I don't have the time to go through that today, but I'll just give you the summary. We spent a good time looking at what it does. And one brief word is that when you have high calcium levels, when the levels are not brought down to normal, it activates two enzymes, that nitric oxide synthase, endothelial nitric oxide synthase, and neuronal nitric oxide synthase. And once these nitric oxide synthase enzymes are activated, they produce nitric oxide, which is important, but again, in excess levels is toxic and oxidative stress. So that's one mechanism that we've actually had evidence for, and which I'm not going to talk about. But we found that PEMCA interacts with the NOS because what PEMCA does, it negatively regulates them. It keeps them undercover. They don't produce a lot of. And we also found that PEMCA interacts with another protein called Cas. And we found that another protein called JAM-A interacts with Cas. So we actually discovered 5 genes and 5 proteins involved in this work. And just to show you that whenever we find something in our mice, we always run to the human. We have a collaboration with the RAD, Reproductive Association of Delaware, that's on this, and we get sperm from them, from men who are undergoing IVF. And we got some sperm from them, and we looked at one of the proteins that's involved in this interaction called JAM-A. Just want to show you, when we get the samples from RAD, we get the motility on them. Here is a patient with 16% motility, and here is one with 54, which is normal. That's a good normal motility. And what you see is that the patient with 60% has no JAM present, active. This is flow cytometry. The blue is the non-specific control, and the red shows you the amount of JAM-A. Sorry, the red is the non-specific, and the blue shows you the amount of JAM-A. When you look at B, you see the left shift, the right shift here, showing you the specific antibody showing you JAM-A. So we had a correlation here between the presence of JAM-A and motility. So as far as humans are concerned, we do know that asthenospermia is the name for reduced sperm motility. That mutations not only in JAM-A, but mutations exist in eNOS, endothelial nitric oxide synthase. We've not yet found heard of a mutation in PEMCA4, but certainly we will, now that we know that we can begin to look for it. So in the fluids of the female tract, we found, I told you we found PEMCA in the fluid of the epididymis, and we found it in the uterine fluid and the oviductal fluid. And in these fluids, the vesicles were identified and shown to have, by immunofluorescence, immunogold labeling and TEM, you can see the presence of PEMCA4 on the vesicles, or oviductosomes, or OVS as we call them, both in the uterosomes as well as in the oviductosomes. And over there, you can see it's not found in the supernatant. It's found solely in the pellet, the cellular part. So we figured if it's found in the oviductal and uterine fluid, then it must be in the uterus. So we looked at sections in the murine endometrium, and if you look at the red signal here, you see that the red signal is found only in the proestrus and the estrus stages, which are the stages that mice will accept males. Mice are not like humans. They only mate when they're in estrus. So what happens with mice is that the female will only accept the males during the proestrus and estrus period. They will not accept the male during the metestrus or diestrus. And so what you see is a correlation of the time they will accept the male with the red or the presence of PEMCA, telling us that there's some relevance in the sperm maturation there. You also see that over here that the when you do westerns, there's marginal levels of the protein at the stage and abundantly expressed at the stage at which proestrus and estrus, at the stage at which sperm will be passing. So we looked at the entire female tract, vagina, uterus, and oviduct. I mean, you can see the red all over it. We found the RNA there, and we found the protein by another way as well. So it's made there, and we did this during superovulation. We can superovulate the animal by injecting them with hormones to have them be in estrus. And when they're in estrus, you can see how everything is all lit up. So again, this is associated with the presence of sperm. We find also that the fluid, look at the levels of the fluid. The oviductal has levels that are significantly higher than the uterine fluid and the vaginal fluid. So it tells us that the oviduct is really important here. And then we are able to show that if you incubate the sperm in the oviductal fluid, you will increase the amount of PEMCA on it. You look at this green shift to the right, and that's after co-incubation, and then this is before, without co-incubation. So the next thing we did was to separate the oviductosomes from the fluid and ask whether if you just incubate your sperm only with the oviductosomes, what impact does it have? And you can see very clearly there's a threefold increase in PEMCA when they're incubated with the oviductosomes, telling us that oviductosomes are necessary and sufficient for the delivery of PEMCA to sperm. What is the mechanism by which this delivery is made? And we, to do this, we labeled our exosomes, with a lipophilic dye, which is red, and we co-incubated the sperm, and what you can see is all these red dots there are oviductosomes, and a fusion mechanism is involved. The membrane, remember these vesicles are membrane vesicles, they fuse with the sperm membrane, and this fusion is the mechanism by which they're able to deliver their proteins or their cargo to sperm. Here we were able to colocalize the labeled oviductosomes and PEMCA, and you can see here that the PEMCA is green and the oviductosome is red, and you can see when they merge there, you have a yellow, telling us that that particular oviductosome is carrying. You might see it better over here, where you can see a sperm head, and you can see this yellow merging there because the oviductosome is carrying PEMCA. This one is all red because it doesn't carry PEMCA, which tells us that all oviductosomes don't have the same cargo. We went on to show that this fusion is triggered by integrins that are on the surface of the membrane of the sperm and the oviductosomes, and it's this ligand pulls the two membranes together. Here is the oviductosomes, here the sperm membrane, here are the ligands, and it pulls them together, and when they come within a certain distance, fusion occurs. So we've actually worked out the mechanism quite well. So what is the impact of PEMCA when it's picked up? Is it still functionally active? And the answer is yes, it is functionally active. Because here we have some wild-type oviductosomes, we have some wild-type sperm, we co-incubated them together, and you see a significant increase. So it isn't picking up dead PEMCA, it's picking up PEMCA that has functional activity. So does the PEMCA that's acquired reduce the calcium levels? And here we use wild-type sperm, knockout sperm, we co-incubate them with wild-type oviductosomes, and we were able to rescue the sperm and bring it back down to a level that's equivalent to the wild type. So not only does it have the activity, but it's able to do the function that it's supposed to do. And of course, the motility levels of a sperm significantly increased when we incubated them over a period of time. So we're picking up a protein that is functionally active. We've also found that these vesicles carry microRNA. That is fascinating. A whole bunch of microRNAs, and there are full differences between A and B. A representing the estrous stage, and B representing the diestrous stage. And one fascinating thing is that we found a microRNA, which is not on this group, called 34C5P. And that microRNA is required for the first cleavage metaphase, which takes place in the oviduct. So what have we learned? We've learned that the oviduct is not a passive conduit as once thought to be. It is actively involved in the reproductive process. It secretes molecules, RNA and proteins, that modulate the gamete function, and once it's delivered by the oviductosomes. One protein I didn't talk about much is the oviduct-specific protein that's involved in fertilization. That binds with the sperm and with the egg and I mentioned the microRNA 34C, and we also know that the oviduct secretes proteins that prevent polyspermy. It secretes protein that causes the zona pellucida to harden, so only one sperm can get to do the job. So it's really a very, very important organ. And so the current art that is being formed today in the absence of the gamete interactions in the oviduct. They use a dish, they use an oviduct. So they don't have all the secretions and all these proteins. And so by studying the oviductal secretions, a number of genes and proteins involving gametes and embryo oviductal have been identified. And knowledge of these molecules that pilot these processes offer great opportunity for improving the success rate of clinical art and patient care. And we've identified oviductal proteins that could enhance sperm survival, particularly for people in agriculture, where they're collecting sperm and freezing down. We need to keep the buffer of the medium in which you keep the sperm is critical for their survival. And if we put some of these proteins in, we could probably increase survival. We could increase the media culture as well for the embryo. But it turns out that embryos and sperm are remarkably resistant towards uptake of exogenous substances in medium. They are very, very resilient. They don't pick up very easily. And so a number of individuals have tried to use nanoparticles as a platform for supplying these things. And the platforms that they've used have been so far inorganic platforms, which are non-biodegradable. Because they're non-biodegradable, they can only be used in the experimental setting so far. But we discovered exosomes, which are biodegradable and which are natural nanoshuttles. And so we think that these exosomes can be used to carry specific molecules for individuals who are missing them. The knowledge gained can be used for individuals with specific deficiencies, and we can also use them when we don't know what the deficiency is. We know what is required, so we can make a little cocktail, add that into the medium, and then hopefully have beneficial effects on increasing this. And so my last slide is to say that the translational impact of discovery of PEMCA4 interacting sperm proteins in the calcium toolkit involves understanding that PEMCA plays a role in integrating nitric oxide and calcium signaling. It's important to note that we, for the first time, have identified two isoforms of PEMCA and showed that they're in all of the journey that the sperm takes: epididymis, uterus, oviduct. And we've identified these genes that not the nitric oxide genes, the Cas and the JAM8, which are partners interacting with PEMCA. So we now have a chance for novel diagnostic targets for acinospermia, which is reduced sperm motility. We can look for mutations in all of these 5 genes for PEMCA4, JAM8, Cas, NOS, and ENOS. We could screen patients for these mutations and then design rational therapy for these specific individuals rather than having a symptom-based approach. But of course, another therapeutic strategy would be to make artificial vesicles, and in these artificial vesicles, incorporate fertility modulating proteins, and those could be used for IUD and for IVF to increase the success rate and to avoid the harmful success of the harmful effects of ICSCI. A number of graduate students have worked on this project. 3 in green are the ones. Amal discovered oviductosomes. Genevieve discovered uterosomes. Rachel showed PEMCA in humans. And Dipti, I haven't told you about her work, but she's got some really fun things that she's just discovered that I want to talk about now. And to mention also my colleagues and collaborators from over the world and my collaborators here at Delaware, and my funding from the NIH, the National Science Foundation, and help from the Reproductive Associates of Delaware. Thank you so much. Any questions that you might have? Yes.
That was really fascinating. If you do get to the point where you can screen people for that, wouldn't that add to the already expensive cost of these procedures of IVF and?
Oh no, the screening is not expensive. No, the screening would be PCR. So what you do is that, because we now do some limited degree of screening for Y deletions. I mentioned earlier on that Y chromosome deletions cause azoospermia or oligospermia, and you simply take a small sample of blood.
Oh,
Is that what you do? Exercise. Oh, I'd be very thin if I were here. Yeah, so that wouldn't definitely add to the cost. And it's compared. This is an in vitro. This is PCR is very automated. You put it in the machine. In the evening, overnight, next morning you have your results. It's not an expensive procedure to screen.
But it would add to the cost, though, wouldn't it?
Well, once you screen, yeah, it would add to the cost, but the therapy that you're going to do is going to be it's not going to be at the same cost of what you're doing, because the therapy that you're going to do, I didn't tell you about. I told you quickly. I wasn't sure. I didn't what kind of audience I had, so I couldn't talk about some of the things that we're doing. But the nitric oxide I told you about, which is toxic. It's a gas, right? And you can pull it through the membrane. And there's a way to have a nitric scavenger to pull it out once you find out, and it's nowhere expensive as some of it. So there are some inexpensive. That's one of the things that you can do specifically for nitric oxide toxicity. For making the models, remember that you're not. It's not going to increase the cost any significant way, because it will be a net decrease when the new novel strategies are used. Yes.
That was really fascinating. Thank you. I have a series of questions. So the PEMCA and the sperm proteins. I think you mentioned the way they were getting into the sperm, but does it need receptors? Are there specific receptors?
Good question. Okay. Good question.
And then, does each sperm have variation in numbers of proteins that will influence, in fact, the mobility, and that's how they are selected?
Excellent question. Excellent question. First question is, most of these proteins are sperm. Most of these proteins, all the I told you about, are made in the testes. So they get some of the testes to start out with. You saw that sperm at the top of I showed you at the top of the tract, it had some on it, right? And in terms of how receptors, how do they get there? How do they, so they start with some at the top, and they get on the sperm by the integrins that I told you about. What are the receptors? You have the integrin receptor, integrin, and there are ligands there that come between the integrin on the sperm and on the oviduct that hold them together. And when they get close enough, the membranes merge, then they oppose that cell is fused. So integrins are the molecular target, the molecular guys that bring them together. And the other question was?
Yes, I was wondering if there are variations.
Great, excellent question. I started my work doing sperm aging in the male. I was a naive graduate student to do my PhD at Western Ontario. And the old female caused Down syndrome. So we wanted to find out if old women have old husbands too. So they could be the fault, kind of stuff. And we studied sperm aging in the male and found that when we age the sperm in the male, the longer they sit in that duct, the more they get on it, the more of the protein they absorb. So sperm population is extremely heterogeneous. When you get a sample of sperm, that have some, in that population, there are some that came into the duct last night because the cauda is the direct source of sperm ejaculation, right? So some were sitting there for 21 days. In mice, they live for 21 days. We checked our mice, they have spontaneous seminal emissions every 21 days. And if you pick up a male, you don't know if he had a spontaneous emission last night or this morning, and then you sacrifice that male and get sperm, and you took up one that's been sitting there you have a totally different, because the longer they stay in that duct, they acquire more. So
they acquire more receptors?
Not receptors. They acquire more of the proteins on them, because the receptors are the integrin that I told you that they have on it. So it took me like almost 30 years to answer the question. As a grad student, why, when you sperm, what happened? We used to age sperm, started at that time with a rabbit. The rabbit is a reflex ovulator. The rabbit only ovulates when the male jumps on her back, right? So it's quite different from humans and mice that have a estrus or menstrual cycle. And when we worked in the rabbit, what we found is when we aged, we could age the male in the rabbit by simply doing surgery, like a vasectomy. We cut the ducts and prevent we cut the epididymis ducts and prevent fresh sperm from going down. And then lo and behold, we got a whole bunch of trisomies and triploides, chromosome abnormalities with age. And that was kind of hard to explain because, as you know, the chromosome numbers are decided way back in the testis during meiosis. So how could you generate extra chromosome? And it was the fact that it was a result of the fact that those sperm didn't have fertilizing competence when they were young. And as they sit in that duct for a longer period of time, it was the accumulation of these fertility modulating proteins that they got that pushed them through. So that was one way of answering why I got a significantly increased frequency of Down syndrome type chromosome abnormalities when I age a sperm in the male. But sitting in those ducts are required for maintaining you have to remember, the sperm without an cytoplasm, they can't fight harmful, they have no machinery for taking care of themselves, protecting them. And it all comes from what they acquire on their surface from the proteins in the fluid which they float around.
I have another question.
Yes.
So you said that obviously PEMCA and the sperm are also produced in the oviduct.
Mm-hmm.
Right? So could a cause of infertility due to the lack of these proteins in the oviduct, so mostly female cause of infertility?
Yes. There are female causes of infertility for a number of genes that are, if you don't have the OVPS, a number of genes, one of the fascinating things in the oviduct, you have cilia that is responsible for the movement of the oocyte towards the egg. And I didn't tell you this, but the female PEMCA4 are fertile. And we just couldn't understand, why are the males fertile when the PEMK4 is knocked out, and the females why are the males sterile and the females fertile? Because what happens is that at high levels of calcium obstructs the cilia, and flagella have the same microtubule structure. Remember the 9+2 in the cilia? And so the same mechanism that's messing up the flagella should be messing up the cilia. And we just wonder what's going on. It turns out that in the oviduct, PEMK1 is turned on, elevated when PEMK4 is knocked out. And that is to rescue the oocytes. So there you go. And there are a number of genes that, when they're not expressed in the oviduct, there is. And we do believe that the expression of PEMK1 and PEMK4 in the oviduct is important. That expression is important for the sperm to actually complete its job. So really, I mean, I've been an andrologist for most of my life, but I didn't realize I wasted all my time with the oviduct, because it's far more fascinating. I'm a late starter.